Welcome. This presentation examines food additives through a systems biology and toxicology framework, mapping how common additive classes interact with biological systems and contribute to downstream health outcomes. Presented by Brian Geniuson, research scientist, this analysis integrates molecular toxicology, cellular biology, microbiome science, and translational evidence evaluation to provide a structured understanding of additive exposure. In this discussion, the reported and scientifically investigated health effects associated with food additives will be evaluated, clearly distinguishing between human clinical data, biomarker findings, and mechanistic research. The analysis begins with the strongest domains of evidence, including inflammatory signaling, microbiome disruption, and metabolic pathway alterations, before examining additional mechanistic and emerging research areas currently under investigation. Food additives represent a broad class of compounds introduced into processed foods for preservation, texture modification, flavor enhancement, and visual appeal. Major categories include preservatives, colorants, sweeteners, emulsifiers, and flavor enhancers. While these compounds serve functional roles in food systems, their biological interactions extend beyond ingestion and into molecular, cellular, and systems-level processes. From a systems biology perspective, additive exposure begins at the gastrointestinal interface. This interface governs absorption dynamics, microbiome interaction, epithelial barrier integrity, and immune system activation. These early-stage interactions serve as primary drivers of downstream systemic effects. At the molecular level, additive exposure intersects with several core biological pathways. Oxidative stress signaling involves reactive oxygen species that influence redox balance, mitochondrial function, and DNA integrity. Inflammatory pathways, including nuclear factor kappa B and cytokine signaling cascades, contribute to systemic inflammatory responses. Microbiome host signaling networks are influenced by changes in microbial composition, affecting metabolite production and immune modulation. Epithelial barrier regulation, particularly tight junction integrity, plays a central role in maintaining permeability and preventing translocation of inflammatory mediators. Endocrine signaling pathways may be influenced by certain additives, although evidence strength varies depending on compound and exposure context. Neuroimmune signaling represents an emerging area of investigation, linking gut activity with neurological and immune responses. Genotoxic and carcinogenic pathways have been investigated for certain compounds, particularly in relation to DNA damage and mutagenesis. It is important to distinguish between mechanistic plausibility and confirmed clinical outcomes. Not all observed cellular or preclinical effects translate into measurable human disease states. Within the empirical evidence landscape, human data demonstrate associations between additive-rich dietary patterns and increased inflammatory biomarkers, metabolic dysregulation, and gastrointestinal disturbances. Biomarker studies support changes in gut microbiota composition and epithelial barrier function. Animal models provide detailed mechanistic insights into tissue-level and systemic responses. Cellular studies further characterize pathway-specific interactions. Computational systems modeling remains an emerging area. Empirical evidence key points. First, additive exposure interacts primarily at the gastrointestinal interface, influencing microbiome composition and epithelial barrier integrity. Second, the strongest currently investigated evidence supports inflammatory signaling, metabolic pathway alterations, and gastrointestinal system effects. Third, many broader health concerns remain supported more strongly by mechanistic and preclinical data than by definitive long-term human clinical outcomes. Key concepts. Additive exposure is system-driven, not isolated. The gastrointestinal interface is the primary biological gateway. Microbiome and barrier interactions are central to downstream effects. Inflammatory and metabolic pathways represent the strongest evidence domains. Dose, duration, and cumulative exposure determine biological impact. Mechanistic findings do not equal clinical outcomes. From a health outcomes perspective, additive exposure has been associated with inflammatory responses, metabolic disease risk markers, gastrointestinal dysfunction, and microbiome imbalance. Certain compounds have also been investigated in the context of carcinogenic processes, although evidence varies significantly depending on additive class, dose, and exposure conditions. Within the validated clinical and biomarker outcomes framework, the most defensible conclusions relate to measurable inflammatory biomarkers, metabolic signaling alterations, and microbiome composition changes. Long-term disease causation remains complex and multifactorial. Conclusion: overall, current evidence supports biologically plausible interactions between food additive exposure and multiple physiological systems, particularly within inflammatory, metabolic, gastrointestinal, and microbiome-related domains. However, translational maturity remains heterogeneous. The strongest support exists at the biomarker and mechanistic levels, while long-term clinical outcome data remain limited and variable across compound classes. Much of the broader health discourse continues to be informed by preclinical and mechanistic findings. Continued high-quality human research is required to clarify causal relationships, dose-response effects, and long-term health implications. Full scientific literature references are available in the video description, and extended reference materials are available by download. This content is provided for educational and scientific discussion purposes only and does not constitute medical advice, diagnosis, or treatment. For continued evidence-based analysis across toxicology, food systems, molecular biology, and systems medicine, consider subscribing.